Name, and you might or might not be familiar with the fact that we are very proud of our football team. I'm talking about American football, and I figured it was just appropriate to introduce our mascot, the Fighting Irish. So my group at Notre Dame studies networks. And why? Well, because networks are everywhere. You can model virtually any phenomenon that you can see in real life with a network. Including cells. So we have these biological networks which typically model relationships between proteins or genes. And if you really represent cellular data in graph theoretic sense, you get something that looks like this. So just by visually inspecting the network, you really can't do much. So we have to develop efficient mathematical and computational strategies and algorithms for studying the data. And that's one of the key focuses of my group. And then secondary interest is, so first, what I mean by efficiently study these networks, I mean how to extract novel biological knowledge from the structure topology of these networks. And then another interest of ours is actually using our methods as well as other groups' state-of-the-art methods in collaborative applications to try to really attack some hard fundamental biological questions. So one of the processes I'm interested in studying is human aging. Why? Well, hopefully all of us would like to live longer and importantly healthier lives. And second, it's very well funded by many agencies, including NSF in the US. NSF will fund your study of aging. It will not fund study of disease. They refer you to NIH for that purpose. So as a young investigator, you have to seek problems that are actually fundable. However, to study human aging experimentally, it's really hard because of long lifespan as well as ethical constraints. So majority of the current knowledge about human aging, which is very limited, has been obtained computationally and not experimentally. And what I mean by computationally is as follows. Basically, you can collect gene expression data at different ages. These are typically collected from different individuals. And then you simply study changes in expression profiles of genes, and if genes are differentially expressed in young versus old age, you predict them to be aging related. So when I talk about ground truth knowledge about aging, I'm really putting ground truth in quotes because it's computational and statistical predictions. That's about it. Another popular approach is not by gene expression analysis, but sequence-based analysis. So namely, in studying aging in model species experimentally is relatively easier compared to doing so in human. So if we know that this particular gene has been experimentally validated to be involved in aging in a model species, then we find its sequence ortholog in human and simply transfer the knowledge between the conserved sequence regions. Now, our hypothesis is proteins and genes interact in the cell to perform cellular function. They don't act in isolation. So why are these current approaches ignoring the important information encoded into the connectivity in the genes, between the genes and proteins in the cell? So we propose two alternative strategies which will actually take the best of each of these. Use the motivation typically done in gene expression analysis to construct, I will tell you what this means, I will go into details on this. You construct dynamic age-specific data and study changes of network structure with age, in the process, importantly, incorporating gene expression data, okay? So it's kind of win-win. And similarly here, instead of aligning sequence-similar regions between different species, align network-similar regions between different species, and then transfer experimentally obtained aging-related knowledge from model species to human between the conserved network regions. Importantly, again, in the process, you can include sequence similarity information while you're constructing these alignments. So again, win-win. And I will focus in more detail on the first topic and just point you to some references that we've done in this context in the interest of time. So the first topic was published in Bioinformatics 2014 paper. And the motivation was as follows. Network research has received significant attention in the last decade or so, but it has research has been done mostly by analyzing static network representations of the cell. And around 2010 or so, it has been recognized that it's need to move towards dynamic network analysis. Now, the problem, unlike in other domains, for example, in social networks, you can actually measure dynamic network structure. You know when each event communication between two people occurs. in cell that is extremely hard due to limitations of biotechnologies. So we have to somehow use the static network data that we have and maybe some alternative dynamic measurements, alternative omics data, and with process of data integration, achieve the goal of computationally inferring the dynamic networks in hope that studying the resulting dynamic networks would give us more biological knowledge and more accurate, that it would reveal more accurate knowledge than traditional static network analysis. And a number of papers has appeared since. These are review papers, not the detailed particular studies, on benefits of data integration, including one of them is in the context of dynamic network analysis. So the general idea is as follows. If this is my static network, well, maybe only these blue genes are proteins. And for just to clarify, we did our study on the case of protein-protein interaction networks, but approaches are, I'm going to present are applicable to any network type, not just other biological network types, but other domains as well. But assume these are genes or proteins that are actually active in the cell at a given age. So maybe we should only focus on subnetwork of the entire network that is active. I will tell you what I mean by active at a given time. And now we have this evolving network where positions of nodes are fixed in this 2D plane. So this node corresponds to, these three nodes are actually the same node at three different time points. So then the question is how do we efficiently, if we have network, let's assume we have network like this, how do we efficiently study changes in network structure with time? So what we did is we took static human protein-protein interaction network. Key results we presented in the paper are from network from HPRD that consisted of the presented number of proteins and interactions. I want to say that in parallel, we also studied the biogrid network, which at that time had about 15,000 edges more. And we did also everything I'm going to show on biogrid network to illustrate the robustness of our approach to choice of particular PPI network source. Then we integrated the static network with aging-related gene expression data. In particular, we based our study on microarray data from 2008 that contains the displayed number of samples and individuals, clearly multiple samples and individuals per ages. In total, we had info on 37 different ages. I want to comment that, again, to illustrate robustness of our approach, we also did exactly the same on RNA-seq data from 2013, and we got quite similar results. So what we did here is, in each sample, we found active genes. What I mean by that is that we measured the intensity p-value of a given gene, which kind of tells you how significant its mRNA abundance is in a given sample compared to other genes of interest. And then we simply took all the active genes in a given sample and our interactions between them. Induced subgraph on active genes, basically. I want to comment that there was no novelty here in terms of network inference strategy. We just used this particular way to do it as a proof of concept. Our goal was to get to networks and then do something fun with the corresponding networks. We have a paper that appeared in Network Science in 2014, which compares network inference in the context of static network research. It compares the different network inference methods, and the key output we get is different methods lead to very different networks, both in terms of network topology and in terms of functional content that is enriched in those networks. So it's a huge, extremely complex problem. We don't believe we have the network inference method for dynamic networks. It's just a very, very simple proof of concept. And I strongly suggest if you're doing network inference and you arbitrarily decide to choose Pearson correlation to correlate genes, expressions levels of genes across different conditions, and you arbitrarily choose threshold of 0.9 to consider edges versus non-edges, well, it might not be optimal way to do so. So once we got to these dynamic age-specific networks, remember there's 37 different networks corresponding to 37 different ages. We first studied global network changes in global network structure with age. And we did 3 different analyses in this context and found no significant change with age. We consider that to be a very good sign. Cell more or less functions similarly with different ages. It doesn't break, so good, right? But then we figured, how about if we zoom into local network neighborhoods of individual nodes and study whether some nodes become more or less central with age? That could indicate that such genes are involved in aging. So I will summarize you that step now. If this is the same toy network I showed you before, for example, we can notice that this particular gene loses connectivity, mainly its degree decreases with age, which could indicate some involvement of this gene in age. And degree is just one of very many possible measures of network position of a node, which we also refer to as node centralities. And we studied 7 different node centralities: betweenness, closeness, clustering coefficient, degree, they are sorted alphabetically, eccentricity, graphlet-based centrality, and k-corness centrality. And what you see on y-axis for each of the centralities, the number of aging-related genes that we predict. These are genes whose network positions significantly change with age with respect to a given centrality measures. Significantly, I'd be happy to collaborate to elaborate on that later on, so feel free to ask me. And also we here present the union of all predictions that we got by taking all the, by combining all the centralities together. And you will in particular note that there's one centrality that captures almost all of the predictions captured by the union of all 7 centralities. This is our graphel-based method. For some reason, the reference does not display. We published that in +1 in 2011. So here's, for network people, quite an interesting finding. What here we show on x-axis is the number of node centralities, and then on, there's 7 possible ones, and on y-axis the number of aging-related genes supported by exactly that many centralities. So what you see here is that the majority of the genes we predict are supported by a single centrality. And as you increase the value on x-axis, you decrease the number of predictions. So there is majority of the centralities are not redundant to each other. So again, if you're a network scientist and you decide, if you're studying networks and you decide to use between a centrality because it's very popular, think twice, because really you might lose lots of signal. You might do better if you consider studying multiple centralities. Okay, so we got a list of predictions. Who cares, right? Well, how do we validate them? Because eventually we want people to go and try to experimentally validate at least the top scoring, the highest confidence ones. So first thing you do, of course, is to make sure your predictions are, I mean, even here when we make predictions, we ensure that they're statistically significant, non-random, but then we performed basically we performed our approach on completely randomized data to ensure that we still couldn't get same or similar predictions. And of course, that works quite well, but as we often know, statistics might be required, but is not necessarily sufficient for biology. So we performed additional, more bio-oriented validation of our results, including the following. So on y-axis, we still see the total number of predictions that we make by taking the union of all 7 centralities. And these are existing ground truth aging-related data sets. First one is actually the gene expression data we use in our study. We didn't do this to validate our genes. We analyzed this, the ground truth data set in this context by measuring of all the predictions that we make, how many of them are in this particular ground truth data set versus how many are missed. So this bar is not supposed to validate our predictions. There's circular argument there clearly, but what it's illustrating is, look at, by using gene expression data, we still get more than 100 predictions on top of that just by using network-based analysis. So networks can be used to complement the knowledge we have by current methods. The other 3 bars are independent ground truth aging-related data sets. Another one obtained by expression data. This is the list of genes involved in Alzheimer's disease. I want to note that the gene expression data that we used was related to brain. So the fact that even in these 2 cases we see significant overlap of our predictions with ground truth data, it's optimistic. This is what is we call sequence age, or formally it's gen age. It's one of the most trusted sources of biological data obtained by sequence analysis. Now, we don't have significant overlap there. I can comment on a follow-up study to understand why that is, but my argument is, well, they are also computational predictions, so who says they are correct, right? So we zoom further into what are the biological processes and diseases enriched in our predictions, and I'm just listing illustrations here. There's probably order of 100 Go terms that are enriched, so for further discussion, please refer to the paper or talk to me, but cell aging, being one of the key aging-related Go terms, is significantly enriched in our predictions, and so are these many diseases, including, for example, these four, and we have managed to find literature evidence that each of these four, this is explicit literature evidence that they are linked to aging. And this is one of the key points I want to make related to using networks to complement insights obtained by gene expression and sequence analysis. There were 6 diseases enriched in our predictions that were missed by each ground truth data set on aging. And for 5 out of these 6, we managed to link them to aging in the literature, which is, again, a very good indicator of our results. 20% of all predictions, so about 100 or so of them, were not found in any of the ground truth data. And we wanted to go into the literature and find whether there's some recent articles linking these predictions to aging. And we couldn't do literature validation on all 100 plus genes. We focused on top 10% of them, okay? And there were 8 genes, top 10% that were not in any of the ground truth data. There were 8 genes, and for each of the 8 genes, we found literature articles, these are the PubMed IDs, indicating directly or indirectly that these genes are indeed linked to aging. For example, these are just slightly paraphrased phrases from the paper. We paraphrase them just to fit the grammar and style of the slides. I will probably want to conclude with this, skipping the network alignment topic in the interest of time. We have applied very similar analysis in alternative domain, that of social network, with important implications to health. We have collected at Notre Dame social network data modeling interactions between people with respect to SMS communication, phone call, email, Facebook, and proximity as measured by Bluetooth over time. And on each node, in previous application, we had one trait, gene is either aging or non-aging, and we managed to distinguish between aging and non-aging genes based on their evolving network signatures. Well, we tried to do the same here for all of these different traits, body mass index, weight, happiness, health, many of them health-related, and each of these network types. So when I show you a colored square, that means we managed to correctly distinguish, for example, between men and women, between people with low body mass index, normal and high, and so forth. And in particular, I'll also show you results that we obtained only by doing dynamic network analysis, only static analysis, and the overlap of the two. And you will notice that the overlap is insignificant. So dynamic network analysis can not only complement existing research on gene expression data or sequence data, but also on static network analysis itself. Now you're going to ask me which one's more accurate, static or dynamic, right? Well, we had a way to validate this because we have a lot more ground truth data in social network research, because you can actually ask people things rather than experimentally try to detect them about genes or proteins. In the cell, I'll conclude in just a second. So in any case, the higher this number, the better, and clearly dynamic network analysis has a lot more higher validation rate compared to static network analysis. I have a follow-up talk on improved methodology. We are going to present that on the last day of the conference in the afternoon, so hopefully some of you will be there. On network alignment, I'm just going to browse through these slides without saying anything about them. Please check out Magna++, our new method for network alignment. In particular, if you're developing a new method, you definitely want to compare against Magna++. Very friendly user interface. Only what bioscientists need to specify is only 2 networks of interest to align. And I'm going to point you to a survey on network alignment we have just published. We have one more talk and 5 posters, so for any technical questions you have about the work I presented, or if the topic of network alignment seems interesting, please come to talk to us. And I would like to acknowledge my group, my 5 PhD students. All of them are here but Yuri. He's doing Facebook internship this summer, so he's quite busy with that. My brilliant undergraduate students, well, actually, both just graduated and will be starting PhD at Stanford in the fall. Most of the work presented here is funded by my NSF grants, and then other projects in my lab are funded by collaborative Google and NIH awards. I'd be happy to take any questions. I know we are short on time. Thank you.
So again, if there are any questions, we should probably move them to after, because we are 1 minute over.